continuing on with relation to the questions that have been coming in Karnataka entrance. The next possible questions that have been coming in right through from the last 10 years include rDNA enzymes. Now, when you say rDNA enzymes, R refers to recombinant DNA enzymes. That is, enzymes which help in joining DNA pieces like ligases, enzymes which are termed as NICs, which can break down nitrogen bases, NICs, as well as we have helicases, which can dismantle the sort of DNA, they can separate the sort of DNA, ligases, and again we have even DNA polymerase. Now, DNA polymerase enzymes are those which go to form the polymer. So, DNA polymerase enzymes are very important enzymes which start the sort of a formation of the DNA during DNA replication. Now, when you take DNA polymerase could be of type 1, type 2, as well as type 3. In addition to this, we also have primase. Primase are special enzymes which are present in cells which act like a primer. They act like a primer, that is, they are the start enzymes. So, primarily a good knowledge of all the recombinant DNA enzymes which are involved in DNA replication, which are involved in transcription, as well as in translation, and in the central dogma. Form essential readings. So, while when we are doing the objectives, we would be checking on the different sort of recombinant enzymes and their sort of roles. The second cluster of questions can also be coming from, so the first is rDNA enzymes. Second cluster can come from Agrobacterium tumefaciens. Agrobacterium tumefaciens is a tumor-inducing bacteria, soil, rod-negative bacteria, and primarily Agrobacterium tumefaciens induces crown gall disease, and the crown gall disease induction is due to the presence of tumor-inducing principle or the TI plasmid. The tumor-inducing plasmid predominantly has been one of the most important discoveries in the 1980s, and we understand that Agrobacterium tumefaciens is a natural genetic engineer. Because it can directly take in an exogenous DNA, an outside DNA, and integrate it into the plant host. Third series of questions which normally can come in is, what are GMOs? When we take GMOs, genetically modified organisms, there are several sort of probable questions which are coming in. One of those includes Dolly the sheep. Then we also have Bacillus thuringiensis, BT plants. Among BT plants, we have BT brinjal, which has the cry gene, which is resistant to aphids. Then lot of questions come from golden rice. Golden rice is transgenic rice. Transgenic rice, whereby rice is fortified. It is fortified with vitamin A. and thereby this is also an example of genetically modified foods and also genetically modified organisms. So when you take three clusters, recombinant DNA technology, agrobacterium tumefaciens, as well as genetically modified organisms, and also we have the concept of the golden rice, which is very important in objective bits, and students should also be knowing about what is the Bacillus thuringiensis plants and how does Bacillus thuringiensis act. Several other questions which normally come in is from genetic fingerprinting. Or DNA fingerprinting, which is related to forensics, or the clue for evidence of DNA in crime or in any paternal duel. For any crime or paternal duel, forensics or DNA fingerprinting is very important. Predominantly, it's also useful for establishing the origin of crops, like the origin of rice. If you take the basmati rice, the basmati rice belongs to India, and it has been proved by means of DNA fingerprinting or genetic fingerprinting. So genetic fingerprinting also gives a sort of a clue or an area wherein we can exactly know about relationships between organisms in terms of their sort of a genomes. The next concept of questions you can also get is from the human genome concept. What is the human genome concept? That is the elucidation of the human DNA. When had it occurred? It had occurred in 2000 as well as in 2001. It had been one of the very important molecular landmarks of the 21st century. So the year, the people who are associated with it, as well as what are the objectives of the Human Genome Project, and how does Human Genome Project form the basis for proteomics, genomics, and metabolics. So knowledge of the Human Genome Project again is the place from which a lot of questions keep coming in. Parallel to these, we have, as implied in the, we have also questions on the wobble. The wobble hypothesis has been proposed by Crick, which says that when we have a nucleotide, when we have a triplet nucleotide called a codon, in the codon, the third nitrogen base is capable of fluctuating. It can hydrogen bond with any other nitrogen base. So the wobble primarily indicates that the third nitrogen base in a codon related to the sort of a tRNA suggests that there could be all possible combinations. So this has been proposed by Crick. So when we elucidate on the genetic code, we will be focusing more on the wobble. Then another series of questions which can come in is from hybrids. Now hybrids are hybrids wherein we have, it is a relationship between the nucleus and the cytoplasm. The nucleus and the cytoplasm together is termed as a hybrid. So while we are dealing with relation to evolution of crop plants, in the context of evolution of crop plants and in the context of medicinally important plants, hybrids play a very important role. Hybrids play a very important sort of a role. Next, classical questions which come in is, what is the powerhouse of the cell? Like as implied, mitochondria are termed as the powerhouses of the cell. Now, why are mitochondria called the powerhouses of the cell? Because predominantly they generate ATP. And it is very, very important that any student primarily understands what happens to the terminal phosphate. The terminal phosphate and how much of energy is stored in the terminal phosphate. So, while we are doing on primarily with relation to electron transport system in mitochondria, we would look in terms of the efficiencies of ATPs which are generated, whether it is 36 or whether it is 38 ATP molecules. Next, questions can also come with relation to who have discovered ATP. If you take Lippmann or even if you take Weismann, the discoverers, the basic discoveries of chloroplast, mitochondria, Golgi apparatus, endoplasmic reticulum, cell walls, nucleus, nucleolus, DNA, it is important that we exactly know about the historical landmarks in cell biology and genetics. The clue to the historical landmarks is from collection of all the Nobel Prize winners. The Nobel Prize winners right till 2013. We have the cargo proteins. The cargo proteins are the docking proteins which carry information as well as sodium and potassium ions from one part of the cell to another part of the cell, for which primarily this year's Nobel Prize has been given to in terms of cargo proteins. So, elucidating of the Nobel Prize winners is very important. The next category you could be getting is primarily on the operon. Lot of questions come on the operon. Now, an operon is a cluster of genes. Which are orchestrated, just like how you play a piano. You have a structural gene, you have an operator gene, then you have a promoter gene and also a repressor gene. So, many a time they could be asking you, what is a repressor? What is an inducer? What switches on the operon? What switches off the operon? What are all the chemicals which can switch on or switch on the operon, especially in terms of the lactose operon? And again, a common question they could ask you is about MULIS, polymerase chain reaction, PCR, wherein we have the amplification of the DNA. A small bit of the DNA can be taken and it can be amplified in different sort of thermic cycles, wherein at each time the DNA can be denatured, separated, and also annealed, that is joined. So, this continuous process of denaturing as well as annealing will synthesize a sort of a daughter DNA. And once you synthesize a daughter DNA, from the daughter DNA, if you add primers, it can easily amplify the sort of DNA and you can know the sort of a nitrogen basis. So, the basic principle of the PCR and who had discovered it, MULIS, is again a very important pointer for any sort of a medical entrance. So, knowledge of the operon would be dealt in detail in a separate sort of a class, wherein we link up chromosome theories as well as molecular biology in the context of biochemistry. So, knowledge, a special concept related to operon is very, very important. Lot of questions come on the operon. What is the lactose? What is the gene called lac operon? How does it function in an organism called Escherichia coli? And when you take the lactose operon, how it is made up of an operator? What is an operator? What is a promoter gene? What are structural genes? What are structural genes? And what is the function of the lactose? If lactose is present in a medium, or if lactose is absent in a medium, what happens to the genes? So many questions come with relation to inhibition, that is, what is the negative inhibition of lactose? What is the positive effects of presence of lactose in a bacterial medium? So operon concept forms the basis for many questions. In addition to this, sometimes they could also be asking you questions on the central dogma. In the central dogma, the most important question which probably they could be asking you is, if given a DNA sequence, what is the mRNA sequence? We know primarily from the ATGC, hydrogen bonds, and the bonding specificity or complementary base pairing. So complementary base pairing exercises need to be done as a sort of a homework. Lot of questions come from, given the sequence of the DNA, what is the mRNA? Given the sequence of the mRNA, what is the sort of a DNA? Then again, on tRNA, many questions come in primarily with relation to what is an anticodon? What is the role of AUG? In a tRNA, what do you understand by the 3' What do you understand by the 5' And how many loops does a tRNA have? How many arms does the tRNA have? What is the DHU loop? How does the tRNA recognize the ribosomes? Questions come on with relation to discoveries of Hawley. Why is the tRNA called the cloverleaf model? Why does it have a cloverleaf model? And tRNA is otherwise termed as soluble RNA. So the tRNA, anticodon, Hawley, loops, 3' 5' directions, arms of the tRNA in relation to ribosomes, what is the function of the DHU loop? These are all very important questions primarily from the viewpoint of an entrance. We understand when we are studying on RNA, many questions come in about the structure and function of the mRNA, the ribosomal RNA, what is the meaning of short-lived RNA, which is very transient, which is completely used up, and again the importance of tRNA. Parallel to this, so they could be asking you on types of RNA, types, structure, related to function, in the context of protein synthesis. Similarly, different sort of an enzymes, like what is a DNA ligase? What is the DNA ligase? Ligase enzyme is one with joins. And again, they could also be asking about lot of questions come on vectors. What are the vectors in genetic engineering? A vector is either is predominantly a virus or a small circular DNA which can carry the recombinant DNA from one host to another host. So basically among the vectors you should know what are the characters of a good vector. What are the characters of a good vector? We would be touching upon the most important feature of PBR 322. Plasmid vector discovered by Bolivar and Rodriguez, and the number is 322. So when we take on vectors, we should exactly be understanding what is a cauliflower mosaic virus, what is a PBR vector, and what is a lambda vector, and what again is a TI plasmid. What are plasmids and what is a tumor inducing plasmid? How they are important with relation to genetic engineering. So we would be checking out on, we would be making a sort of a table saying which are all good vectors. So vectors are small vehicles which transmit DNA. Either they could be taking small uploads of DNA like say 10 to 20 kilobase space, or they could be taking big uploads of the DNA like 10,000 to 20,000 kilobase space. So vectors are very, very important, and when you take a sort of a bacteria, in a bacteria if you take Escherichia coli, you got a circular DNA, and along with the circular DNA you always have a sort of a plastid which is available free. So in the case of Agrobacterium tumefaciens you have a TI plasmid. Even in the case of an Escherichia coli, if this is the DNA, always there is an association of a plasmid. This is a plasmid. Now the plasmid can take in a sort of a, it has an ORI, it has an origin, and it has got several flank regions. We have a left flank and a right flank, and when we have a left flank and a right flank, between that, due to the presence of restriction enzymes, they could also be asking you what are all common restriction enzymes. Restriction enzymes are restriction endonucleases, which can cleave this sort of a DNA at a specific site. So when they cleave the DNA at a specific site, or when they cut the DNA at a specific site, a space is created into which the foreign gene can be inserted or it can be integrated. And the foreign gene which is integrated within the sort of a with the help of the restriction enzyme gets into a plasmid, and the plasmid can move from one host to another host. Like it can move from a bacteria to a plant cell, or from a bacteria to another bacteria. Thereby these are all primarily termed as the tools in genetic engineering. Parallel to the tools, you should also be knowing about the techniques. So in techniques, we have gene cloning. What are all the gene cloning techniques? What is the importance of PCR? What is the role? How do you get restriction enzyme digestion? So most important techniques which they ask you primarily is southern blot. We have southern blotting, and again northern blotting. Parallel to this, we also have, they could also be asking you about polyacrylamide gel electrophoresis, PAGE. Polyacrylamide gel electrophoresis is a very important sort of a tool for separating the different proteins, as well as separating the different sort of a biological compounds on the basis of their molecular weight, on the basis of their migration through a solvent, especially when the molecule is amphoteric and when it is charged as plus and minus. So for separation of different DNA components, different RNA components and protein components, you primarily use the technique which is termed as polyacrylamide gel electrophoresis, and they could be also asking you about techniques of southern blot and northern blot. So these are the basic different sort of techniques which are used in recombinant DNA technology. Even with relation to chromosomes, they could be asking you like what is VNTR? VNTR, that is when you take the T, that is the telomere. On the telomeres, how many repeat sequences are present? how many repeat sequences are present. So a good knowledge about what is a telomere, what is a centromere, and what is the importance of heterochromatin, and again what is the role of euchromatin. So knowledge of heterochromatin, euchromatin, VNTR, telomerase and also centromeres and repeat sequences are extremely important. Parallel to this, they could also be asking you questions on which are all the sort of proteins which bind to the repressor of an operon. When you take an operon, a cluster of genes, it has always got a repressor. So to the repressor, which are all the sort of proteins which can bind? Which are all the proteins which can bind? Many questions come from this. When you take the genetic code, they could also be asking you what are termination codons. So the actuals of how the questions are coming in a competitive examinations is being the subject of discussion down here. They ask you what are termination codons. Can you name them? What are their functions? When are they formed? When are they formed? And when you come back to restriction enzymes, when you take restriction enzymes, common restriction enzymes which primarily are termed as TAG as well as BAM. Then you got HIND3, BAM H1, and again you have ECOR1. These are all restriction enzymes. So many a times they ask you the names of the enzymes and what are the names of the sort of microorganisms from which they have been isolated. Like TAG polymerase is a very important enzyme, and Escherichia ECOR1 from Escherichia restriction enzyme 1, it has been isolated from which sort of a microbes. So a good knowledge of bacteria, fungi, algae and viruses which are sources as restriction enzymes. So it's a sort of a test wherein you're thinking in terms of biotechnology in relation to microorganisms. Questions which have also been coming is Malus with relation to polymerase chain reaction. Then Franklin Condret, who has been associated with the discovery that RNA is a genetic material in tobacco mosaic virus. And again Barbara McClintock, who has been associated with jumping genes or transposons. Transposons are transposable elements. They are transposable mobile elements which are present in maize. In maize, we have genes which are movable. So these have been discovered by a scientist whose name is Barbara McClintock. Again, a very important scientist is Franklin Condret, and again Malus. So this primarily indicates that names of important scientists and their discoveries form the basis for many of the sort of questions of the objective questions. While studying transmission genetics, that is classical genetics, as well as when you study modern genetics deals with linkage and crossing over. It's important we also study molecular genetics. Because molecular genetics forms the basis for evolution. So they could be asking you many questions on blood groups, Landsteiner, who is associated with blood groups. Transmission genetics, they could be asking you questions on Morgan, on sex linkage, as well as on Mendel. In molecular genetics, they could be asking you primarily questions from the operon. That is, we have Beadle and Totem. And again, in molecular genetics, they could also be asking you questions on Corona. You have Corona, who is associated with the artificial synthesis of the gene. That is, in vitro synthesis of the gene. And again, on operon, they could also be asking you on Jacob and Monod. Beadle and Totem are associated with Neurospora genetics. Which deals with transmission. Then again, in evolution, in relation to genetics, they could be asking you the name of the scientist of Wallace. Wallace is very, very important in terms of evolution. So, many questions come out with relation to names of scientists, terminologies, structure, function, relationship, as well as all Nobel Prize winners in genetics. And when we're dealing with Nobel Prize winners in genetics, it is a mix of cell biology, genetics and molecular biology. In protein synthesis, many times questions come on what is the peptide bond? What is the peptide bond made up of? How is the ATP bonding? If you take adenosine triphosphate, how is the last terminal phosphate bond? What are the translation factors in eukaryotes? What is amino acyl site? What is peptidyl site during protein synthesis? And again, what is an amino acyl peptidase? Many questions also ask you what is chain initiation, chain elongation, how does chain elongation and how does chain termination occur during protein synthesis? Again, they could ask you the sequence of the operator genes in an operon. So important here is you should exactly know the steps in chain initiation, elongation and termination in terms of consequences. How is this? What is the start signal? What is the stop signal? What has been derived from these particular steps and how they are linked together. From the areas of DNA replication, protein synthesis, as well as gene regulation and genetic code. All the four areas, there are several sort of questions coming in. Common questions which come in are, what is an Okazaki fragment? Okazaki fragment occurs during DNA replication. During DNA replication, short segments of DNA which are synthesized are termed as Okazaki fragments. And these are short sequences which are formed on the lagging strand. During DNA replication. they require several primers. They require several primers. This is a primer, this is a primer, and all these primers anneal, and thereby the short pieces of the DNA become long and they go to form the sort of a daughter DNA. So many times questions come as what is an Okazaki fragment? And again they could be asking you what is the non-transcribed region. The terminal sequences are termed as the non-transcribed region during DNA replication. Many a time they could also be asking you on what is cDNA. What do you understand by cloning DNA? The new clone which has been formed is primarily termed as cDNA. And questions also come in like what happens when the first base in a nucleotide gets knocked off by means of mutation. So by mutation, whenever there is a mutation, if we have a sequence of nitrogen bases called A, B and C, and if B gets knocked off, then you have the formation of A dot C, and the triplet codon here shifts to D. Instead of A, B and C, the codon, the codon here shifts to form another codon here called ACD. This sort of a mutation is termed as frame shift mutation. So frame shift mutation is an example of point mutation. So the loss of either the first or the second or the third base results in the formation of a frame shift mutation. So questions do come in from even frame shift mutations, what is cDNA, what is a non-transcribed region, and again what are Okazaki fragments. Even from types of DNA, questions ask you about the characters of the A DNA, the B DNA, as well as the Z DNA. How is it helically turned, right-handed or left-handed, and how many nitrogen bases are present within the sort of a DNA strand. Many a time they also ask you about cDNA paradox. That is, how is the nitrogen basis stable despite modifications? And given a coding sequence, they could also be asking you what is the amino acid sequence. So it's very, very important that we understand the genetic code, we understand the triplet nucleotide, and commit to by heart the codon sequences and the respective amino acid sequences. So this forms a very important basic MCQs can come in from here. Questions come in from discovery of transformation, Griffith experiment, discovery of transduction by Zender, discovery of conjugation, which has established sex in bacteria. There are several questions which come on how does the DNA anneal or join. How is the annealing of DNA occurring? And again, how does the DNA unwind? Parallel to this, they could be asking you what is the role of DNA gyrase. What is primarily a sort of a polymerase? What is a polymerase? And again, what are all the enzymes which are responsible, which have a major role in DNA replication? So a table about the different sort of enzymes like helicase, gyrase, topoimerase, primase, ligase, and primarily the gyrase is very important. And many times questions also come from paternal crimes related to DNA fingerprinting. We are dealing with objective bits, like when you take question number 1. Question number 1 says, in his experiment on the chemistry of DNA, Chargaff estimated the base composition of the human sperm and found 31% adenine and 19% guanine. We have 31% is adenine and 19% is guanine. 19% is guanine. The quantity of cytosine in the DNA of the human somatic cell, we are asking about the quantity of cytosine in the human DNA. How much amount of cytosine is present in the human cell is likely to be, the choices are A is 19%, B is 38%, C is 31% and D is 62%. Now we understand always the guanine content shows a base equivalence to the cytosine content. Hence the correct answer is A, which relates to 19%. So this problem deals with Chargaff's rule of base equivalence in the DNA. And notably, you got to understand that guanine and cytosine occur with the same sort of percentage. Hence, the choice answer is A. Now we go to the problem number 2. In problem number 2, it says the DNA molecule in Escherichia coli is heavy due to presence of N15 and is allowed to replicate in a medium containing N14. After one generation of replication, the daughter molecules could have A, will be of similar density but will differ from that of the parent, B, will differ in the density from one another and also from the parental DNA. The third choices will have the same density as that of the parental DNA, and D, they will differ in density from one another but will resemble the parental molecule. We understand that this question relates to the experiment of Michelson and Stahl. Michelson and Stahl had conducted an experiment related to semi-conservative replication. Semi-conservative replication of the DNA. So they had taken Escherichia coli, E. coli, and in E. coli they had taken the sort of DNA, extracted the DNA, and after extracting the DNA, they had labeled it with N15, is radio labeled, and they grew the DNA, they put it in a sort of a medium in which you have N14. So normally what happens is, when you have the N15 DNA, which includes both the helix will separate, and one will form A helix, another will form the B helix. A and B helix both are labeled with N15. So thereby both the helix will be similar in density. However, they differ from that of the parent because the A helix in turn, one will be N15. Along with that, you will have a newly synthesized strand which is N14 from the medium. Thereby, they differ from the parent. So parents will have heavy radioisotopes N15, whereas the first generation will have a cluster of N15 as well as N14. Hence, the answer for question number 2 is answer is A, which states they are similar in density but however what happens is they differ from that of the parents. They differ from the parents. Question number 3 asks how many nucleotides are present in one spiral of the B DNA. Now B DNA is a common DNA. It is a right-handed helix. It is twisted along a perpendicular axis, and in one spiral it is primarily made up of 10 nucleotides. There are 10 nucleotides, or there are 10 steps which are formed due to hydrogen bonding, and there is a pairing between a purine and a pyrimidine. So in one spiral there are, the correct answer is 10 nucleotides, and 10 nucleotides, answer is B. Which indicates this presence of 10 nucleotides. Question number 4 asks what is a nucleoid? What is a nucleoid? A nucleoid, choice A says it is a single inactive nucleus with a double-stranded DNA and a protein. Choice B says it is a nucleus without a karyotheca and a nucleolus. Choice number C says it is a chromosome associated with proteins. And choice number D says it is a group of chromosomes associated with proteins. Nucleotide is normally seen in bacteria. It is also seen in prokaryotes. It does not have a nuclear membrane. Nuclear membrane is absent, and there is no distinction into the nucleus, and again there is no nucleolus. So nucleolus is absent, nuclear membrane is absent, and nucleoid primarily refers to naked DNA. Which is not complex with histones. When you take eukaryotic cells, when you take eukaryotic DNA, in eukaryotic DNA, the DNA is held by histones. Histones act like clips. Like how we hang clothes. So the presence of histones is a characteristic feature of eukaryotic DNA. First thing is, because histones are absent, it is a prokaryotic DNA. And in a prokaryotic DNA, the common feature is it does not have a nuclear membrane. Hence, there is no presence of karyotheca. Hence, the correct answer to this 4 is it is a nucleus without a karyotheca and a nucleolus. Hence, the answer is B. Next, we go to question number 5. Question number 5 asks, nucleoprotein in a cell are synthesized. The nucleoproteins are synthesized in the nucleolus, or is it synthesized in the nuclear membrane, or are nucleoproteins present outside the nucleus, or are they present in the nucleoplasm? Now, nucleoproteins are normally assembled in the cytoplasm. And they reach the nucleus after protein synthesis via the nuclear membrane channels. Later on, the nucleoproteins get clustered or organized inside the nucleolus and inside the nucleoplasm. So predominantly their pattern of the question is, the question asks you where they are synthesized. They are always synthesized outside, because it is formed during protein synthesis. Hence the correct answer is C. C is the correct answer, which states they are synthesized outside the nucleus. Question number C relates to discovery. Nucleic acid was isolated as nuclein for the first time. When you take nucleic acids, it has been isolated as nuclein for the first time. The choices are, is it Koch, Fleming, Altman or Mister? The correct answer is Mister, and the answer is D. We go to question number 7. DNA structure was studied by X-ray diffraction technique by A is Watson, B is Wilkins and Franklin, C is Crick, and D is Watson and Crick. As implied, when you take DNA, diffraction studies or study with X-rays in terms of the DNA sample was studied by Wilkins, and Franklin, Rosalind Franklin. So Wilkins and Rosalind Franklin are associated with X-ray diffraction studies of the DNA, and they have received the Nobel Prize along with Watson and Crick. Hence, for question number 7, the answer is B. Whereas the other choices, when you take only Watson, primarily is associated with nucleic acids, while if you take Crick is associated with central dogma, and when you take Watson and Crick, they are associated with the discovery of the double helical model of the DNA. So Rosalind Franklin's experiment primarily had given proof to state that the DNA is helical. And it had given proof for the diameter of the DNA as well as the geometry of the DNA. And it had been supported by the complementary base pairing. As proposed by Crick. Hence, for question number 7, the right answer is B. Question number 8, the answer is primarily direct. DNA can be tested by means of Schiff's base, because it is acidic. Hence, the answer is B. B is the answer. Question number 9. It reads, which of the following statement is not consistent with the double helix model of the DNA? What is a non-consistent statement? Statement number A says density of DNA decreases on heating. Definitely, when you take the DNA and when you heat it, it denatures, and when there is denature, there is a breakage of the hydrogen bonds, and when there is a breakage of the hydrogen bonds, the adenine, guanine, cytosine and thymine fall off, thereby the density decreases. So it is a correct statement. B bit says always adenine and thymine and cytosine and guanine are present in equal terms. So even B bit is consistent. Whereas when you take bit number C, the amount of A plus T oblique C plus G is not constant in different organs of a species. Whereas here we understand, when you take the Chargaff's rule, according to the Chargaff's rules, we easily understand that always A plus T A plus T and C plus G is always a sort of a constant for a species. Is always a constant for a species. And D, it says according to X-ray diffraction pattern, it's a double helical structure. Even this is a sort of a valid statement. Whereas when you take answer number C, answer C, it says that the amount of adenine, thymine and guanosine and cytosine is not constant. It says though the correct answer is constant, but the bit says it is not constant. Hence, this is non-consistent among all the 4 choices. Hence, the correct answer is C. Question number 10. The 2 strands of the double helix. The 2 strands of the double helix are, choice A is they are coiled side by side. B is they are twisted around each other like 2-stranded ring of a rope. Then choice number C is they are twisted around each other like the strands of 2 strands rope with free ends. And D, they are not coiled structures. When you take the DNA double helix, as you understand, they are strands, they are double stranded. One has a 3' free end and the other has a 5' free end. And because of the presence of the free ends, 3' and the 5' ends, they have an hydroxyl end which is free, and thereby they also show opposite polarity. So due to the presence of hydroxyl free ends, so they are twisted, they are 2 strands, but again the most important point here is they have free ends. Free ends are essential because primarily it balances out the sort of a DNA within the sort of a nucleus, and it also enables it to be more acidic so that it is more reactive even along the major and the minor groove. So the correct answer to this is C. Question number 11 again relates to discoveries. The Nobel Prize in 1968 for the determination of tRNA base sequence. When you take the tRNA base sequence, 1968 was awarded to the choices Nirenberg, Korenberg, Korana and Hawley et al. Now, Hawley et al. Relates to other scientists. He is primarily associated with the tRNA structure. He is associated with the clover leaf model. So he primarily proposed, when you take the base pairs, you have a 3' end and again you have a 5' end. The tRNA has got arms, then again it has got loops, it has a DHU loop and it has an anticodon. And the tRNA is very specific. It is very specific to a specific sort of an amino acid, to any sort of an amino acid. Whereas the other choices, when you take Nirenberg is associated with synthesis of the RNA. Korenberg is associated with again artificial DNA synthesis. Korana for in vitro synthesis of the RNA. So the right answer is Hawley. Hence the choices answer is D. Question number 12 relates to methodology. If one wants to obtain precise information regarding the exact time and location of synthesis of a new DNA, which of the following methods would be very effective for such a study? We are looking in terms of DNA synthesis, we are looking at new daughter DNA, new daughter strands getting formed. That is, when a DNA unwinds, it synthesizes a new strand and again it reforms. It unwinds, so there is synthesis of a new strand, and later on there is reformation of the sort of a DNA. So the choices are A, isolation and extracting the DNA after regular intervals and estimating the DNA amount. We are not estimating the DNA amount. B is using radioactive precursors of nucleic acids. If you use a radioactive precursor, we only exactly know what is the pattern, but the answer it asks is what exactly is the method of DNA synthesis. C, carbon dating relates to fossils. So the only way which is an evidence for exactly knowing the location and the time of DNA synthesis in a cell. In a cell, we are looking at location and we are looking at time of synthesis, wherein we take a sort of a time-lapse photography. Time-lapse photography, or we do a sort of a pulse photography. And for pulse photography, along with location in the nucleus, or in other components, the correct way of analyzing this is by use of D. Answer is electron microscopy. Electron microscopy. So by an electron microscope, you can exactly visualize. You can visualize synthesis, that is if this is the DNA and these are the ribosomes. You can easily visualize the synthesis of daughter strands. The synthesis of daughter strands is in real time. You can exactly view through by use of an electron microscope. So EM studies primarily are responsible for knowing the time, location and time of DNA synthesis. Hence, the answer is D, electron microscopy. We go to question number 13. In a double helix DNA molecule normally found in plants and animals, each successive base pair in a stack turns A choices 36 degrees in the clockwise direction, 36 degrees in the anti-clockwise direction, C 360 degrees in the clockwise direction totally turned, D 360 degrees in the anti-clockwise direction. Normally when you take a normal double helix, when you take a DNA molecule, the DNA molecule in plants or a normal DNA molecule is a right-handed helix, right-handed helix, and it is turned clockwise. It is turned clockwise. It is turned clockwise, and the angle at which it gets turned is 36. It is at 36 degrees. So 36 degrees in a clockwise direction, especially when it's turned on the right. Hence the prime answer is A. The choice is A. Question number 14 says, which is the smallest RNA? RNA can be calculated on the basis of the Swedberg's constant, as well as its molecular weight can be determined by Dalton's. In terms of size, when you take the RNA, we have the messenger RNA, which is quite long. Then we have the ribosomal RNA, which is dense. Then we have the tRNA. The tRNA is also called soluble RNA. It is called soluble RNA. It is called the soluble RNA. And later on, we have the genetic RNA. The genetic RNA refers to the RNA which is the genetic material in tobacco mosaic virus. So the smallest RNA which has got a very high turnover turnover in terms of metabolism. High turnover in terms of metabolism relates to the tRNA. Relates to the tRNA. Hence, the answer is, you got, is C. Correct answer is C. The smallest RNA is a tRNA, because even in time it just fades off. So metabolically it's more active. It's also metabolically more active. Question number 15 asks, the person awarded Nobel Prize for synthesis of RNA 1959. Synthesis of RNA. Especially when you say in vitro synthesis, that is in a test tube. If you take primarily, the correct answer is Ochoa, O-C-H-O-A. Ochoa is a scientist who had been working along with several other associates, and he was the person who did RNA synthesis in vitro. Hence, the right answer is A. Right answer is A. Whereas the other choices are Korenberg is related to DNA synthesis, Nirenberg also DNA synthesis, Corona all by himself did not do RNA synthesis. The correct answer is Ochoa. So the answer is A.